the heart. Lecture will focus on electrical conduction, the cardiac cycle, and how heart function is regulated. Cardiac muscle is a unique muscle tissue. Within the heart wall, the tissue is found in a layer called the myocardium. While there are no fascicles like you'd see with skeletal muscles, cardiac muscle cells are arranged in bundles. Within the superior chambers, atria, the cardiac muscle cells wrap horizontally around the chamber, then move down and twist around the inferior ventricles, the inferior chambers called ventricles. This assists with moving blood through the chambers. The lower chambers, ventricles, squeeze blood like you would twist a dishcloth to squeeze the water out. The myocardium is fed by typical blood vessels, but in the heart, these vessels have structures called anastomoses. We build them as we age. Anastomoses, noted as the dotted lines on the heart diagram, serve as detours, moving blood in and out of the myocardium through multiple pathways. If one is blocked, the myocardium may still be supplied by blood flowing through another pathway. We make or build anastomoses as we age. One theory is we have tiny micro heart attacks as we age, and this may be the response to that. The heart's function is to beat. In doing so, it creates pressure changes that move blood through the chambers and blood vessels. The beating is controlled both extrinsically, outside the system, and intrinsically, inside the system. Extrinsically, the heart is controlled by the nervous system. Nuclei in the medulla oblongata called the sympathetic cardioacceleratory center increase heart rate, while other nuclei in the medulla oblongata called the parasympathetic cardioinhibitory center decrease heart rate. Interestingly, the heart has parasympathetic tone, meaning the heart rate is always a little decreased. The sympathetic nervous system can override this tone. How does it work? Action potentials. You'll learn later the specifics. Valves prevent blood from going backwards in the heart. The tricuspid and bicuspid valves prevent backflow from ventricles to atria as the ventricles are squeezing. The semilunar valves prevent backflow from the artery into the ventricle. As long as valves work, blood moves forward. Heart sounds are the sounds of these flaps closing. The LUP is closure of tricuspid and bicuspid valves. The DUP, closure of semilunar valves. You first learned about cardiac muscle tissue in Bio 2050. These fibers contain intercalated discs, endomysium, and function as a syncytium, which means the cells are interconnected, so they form or function like one big cell. Cardiac muscle cells are innervated by collections of self-depolarizing cells. They contract as a unit thanks to their intercalated discs. They utilize the sliding filament mechanism that skeletal muscles use, and they have an extremely long absolute refractory period. This long refractory period helps ensure that cells in the bundle function as units instead of becoming disorganized. Cardiac cells don't act by themselves. You should take a moment and pause right now. Try to refresh your knowledge of action potentials from Bio 2050. Cardiac muscle tissue is controlled intrinsically by self-depolarizing neurons. These neurons fire pacemaker potentials, allowing them to self-depolarize in a regular pattern. What's different about pacemaker potentials? They reach threshold with sodium, depolarize to peak with calcium. Reach peak at 0 millivolts instead of plus 30 millivolts, and do not hyperpolarize. Their solubility to sodium and lack of hyperpolarization ensures that the cells will easily reach threshold on each cycle. There are specific structures in the heart that start the electrical signals using these special potentials. Again, I suggest you pause here and review the differences between a regular action potential and these pacemaker potentials. Once pacemaker potentials are generated, they travel through these structures. This here is the intrinsic conduction system of the heart. The sinoatrial, or SA node, is located in the septum between the two atria. Pacemaker potentials are generated here, which is why the SA node is known as the pacemaker. Then the action potentials move through the atrial myocardium. At the same time, some potentials move to the AV node. The atrioventricular node is known as the gatekeeper. The AV node delays the potentials for a brief moment. The delay is needed to give the atria time to fully depolarize before controlling ventricles. The next structure, bundle of His, is found in the septum between the ventricles and is a branching point. Then the action potentials move through the equivalent of motor neurons, through the ventricular myocardium, through the Purkinje fibers. Again, I suggest you pause and review these structures. Action potentials are generated in the SA node. Really, they're pacemaker potentials. Then they travel to the AV node where they are delayed so that the atria can behave separately from the ventricles. Then they travel to the bundle of His where they separate, and finally through ventricular myocardium. This diagram shows the locations and pathways of this intrinsic system. Note the locations of the SA node, the AV node, the bundle of His, which has recently changed its name, and the Purkinje fibers. Imagine watching these action potentials move through the heart. An ECG or EKG shows us how pacemaker potentials are generated and how they move through the intrinsic system. Note deflection waves on the diagram. These are electrical events, depolarization, repolarization. Mechanical events, contracting, relaxing, occur in the flat lines that follow the waves. P wave. Pacemaker potentials are being generated at the SA node, and the resulting action potentials are moving through the atrial myocardium. This electrical event is known as atrial self-depolarization. The atria will contract in the flat line that follows. QRS complex. Action potentials are now moving through the ventricle. This is known as ventricular depolarization, and the ventricle should contract in the flat line that follows. Note, the QRS complex and the P wave are out of phase, meaning the QRS complex happens after the P wave. This time shift is because of the functioning gatekeeper AV node. Imagine what would happen without the functional AV node. Action potentials would not be delayed, and the P wave and QRS complex would be too close together. Now, at the same time of this event, the atria are repolarizing. We do not see this in the QRS complex because the ventricles are too large and their event covers up the atria's event. T wave. Action potentials are going away in the ventricles. This time is ventricular repolarization, and the chambers will relax in the flat line that follows. You will learn to calculate data for intervals, rates, and segments in the lab. For now, these data points tell us the story of the patient's heart. We can measure how strong the electrical event is, if the timing between them is correct, and even how long it takes for the chambers to fill with blood or eject blood. Each of these data points has normal range values. Take some minutes now to review the electrical events taking place during the 3 deflection waves. This is not a clinical class, and you will not be expected to make diagnoses from EKGs. However, we do not need to be clinicians to spot a normal sinus rhythm versus abnormal rhythms. A normal sinus rhythm. You can see the 3 waves happening at regular intervals. See the tall R, the P just before it, and the T just after. B, abnormal, junctional rhythm. Note the lack of P waves from a non-functional SA node. C, second-degree heart block. Note the extra P waves and way too long P to R interval as the AV node and bundle of His malfunction. D, ventricular fibrillation, non-functional SA and AV nodes. The ventricles are simply twitching and have no time to fill. Blood is not moving through the heart or the body at this time. VFib must be treated immediately with a defibrillator or CPR, or the person will die. The electrical cycle of the heart controls its mechanical cycle. The cardiac cycle is the interplay of these mechanical events. When a chamber is relaxing, it is in diastole. Relaxing chambers are no longer squeezing. The pressure of blood inside drops, and this allows blood to enter. When a chamber is contracting, it is in systole. Contracting chambers are now squeezing on the blood inside. This increases the pressure of blood inside the chamber, and eventually the pressure is high enough to open a valve. At that point, the chamber will eject the blood out of the chamber into the attached blood vessel. This alternating squeeze and relax, or systole and diastole, create pressure gradients between the chambers, and blood moves. While the atria are in diastole, the ventricles are in systole. Make a model. The top hand represents the atria. Bottom hand represents the ventricle. In this picture, while the atria is relaxing and filling with blood, the ventricle happens to be contracting and ejecting blood into its vessel. If you switch the hand positions, you'll see that while the atria is contracting to move blood, the ventricle is now relaxing and filling with that blood. Note, the atria are thin-walled and their force of contraction only moves a small percent of the blood at best. The main chamber driving pressure and therefore blood movement are the ventricles. Overview. Atria depolarizes, then contracts and ejects blood. Atria repolarizes and relaxes and fills with blood. Out of phase, the ventricle depolarizes and contracts, ejecting blood, then repolarizes and relaxes, filling with blood. Focus on the ventricle. During systole, action potentials arrive. The myocardium depolarizes and the myocardium contracts. The ventricles are now squeezing on the blood inside, and pressure on that blood within the chamber rises. Atrioventricular valves close. Ventricles continue to squeeze and pressure continues to rise. Eventually, pressure gets high enough to open the semilunar valve and blood is ejected. During diastole, action potentials disappear. The myocardium repolarizes and then the myocardium relaxes. Pressure falls inside the chamber. The semilunar valve closes. The ventricle continues to relax and pressure continues to drop. Then the AV valves open and blood enters. Focus on the atria. Remember, the atria are thin-walled, so it is really the ventricles driving valve and pressure changes. Otherwise, the change in pressure is similar for diastole versus systole. Repeat this cycle several times out loud until you've got it memorized. Some terms. Isovolumetric contraction. The period of time in systole when both valves are closed. So the volume of blood does not change, although the pressure continues to rise. Isovolumetric relaxation. The period of time in diastole when both valves are closed. So the volume of blood does not change, although pressure continues to drop. Dicrotic notch. Unique to the aorta. Change in aortic pressure when aortic valve closes. Pressure drops, then rises again. This graph shows changes in pressure in chambers and vessels. The dicrotic notch of the aorta is visible. Terms. Note the definition of each posted term on this slide. Also know the formula. These will be repeated in the lab. Cardiac output is mils blood pumped by a ventricle in 1 minute. Heart rate, number of beats in 1 minute. Stroke volume, milliliters of blood pumped by a ventricle in 1 beat. Cardiac output is equal to heart rate times stroke volume. If you take maximal cardiac output and subtract cardiac output at rest, you'll have cardiac reserve. In short, cardiac output is the amount of blood your heart can move through your body basically in 1 minute. Stroke volume and heart rate are continually changed to modify cardiac output in our bodies for homeostasis. Stroke volume is the difference between end diastolic volume and end systolic volume. What does that really mean? Think of it this way. The amount of blood moved by a ventricle in 1 beat, stroke volume, is equal to the total amount of blood it filled up with minus the blood that was there before it started to fill. Again, amount of blood moved by a ventricle in 1 beat is equal to the amount of blood it filled up with minus the blood that was there before it started to fill. EDV is how much blood entered the ventricle when it relaxed, how much blood it filled up with. ESV is how much blood is left after it contracts, or how much blood was there before it started to fill. Should understand what these two terms mean, EDV and ESV. The myocardium has a unique ability to locally respond. For example, if you've been sitting for a while, the amount of blood entering your right atrium is not that high. Your ventricle is accustomed to that volume. Now, get up and run around your house for a few minutes. This increases the blood returning to your right atrium. The first couple of times this happens, that means extra blood enters the atrium and ventricle and stretches the wall. That stretch is a trigger for the myocardium to contract with greater force on its next cycle. This increases the pressure and helps push the extra blood out of the ventricle. The stretch, called preload, the increased force of contraction, is called contractility. The law of how this works is known as the Frank-Starling law of the heart. Again, extra blood from exercise stretches the right atrium and right ventricle. That is a trigger for the right ventricle to squeeze harder to push the extra blood out. The stretch is called preload. The force of contraction is contractility, and this story is the Frank-Starling law of the heart. Afterload. Afterload is what the left ventricle has to push against. It is related to the amount of blood in the aorta. A diagram of the Frank-Starling law of the heart again. Add extra blood, you will preload the heart and stretch it. That results in greater contractility. The ventricle squeezes harder. It increases stroke volume. Afterload. Too much pressure in the arteries. The ventricles can't push as much blood into that high pressure artery. Blood backs up into the ventricle. You will decrease contractility. If we need to change cardiac output for homeostasis, we do so by changing either heart rate or stroke volume. Remember the formula, CO equals heart rate times stroke volume. There are many ways to change each. This graphic lists some of the ways to modify cardiac output extrinsically, and we'll discuss each in turn. Sympathetic nervous system increases heart rate. The neurotransmitter norepinephrine opens calcium channels so that cardiac cells depolarize faster. This is a positive chronotropic factor. Parasympathetic decreases heart rate. That would be cranial nerve 10, the vagus. Its acetylcholine opens potassium channels so that cardiac cells repolarize. This is a negative chronotropic factor. In fact, the heart is under constant slight inhibition by cranial nerve 10, the vagus. That's called vagal tone. Take some time to stop here and review how the sympathetic and parasympathetic neurotransmitters change heart rate. Now I want you to take a stab at it. Think about these questions. Try to predict what happens to heart rate if you added calcium, blocked calcium with a channel blocker, added potassium, added epinephrine. Post your response in the AMP Cafe discussion board. Only there can you find the answers. pH is a driving force for changing cardiac output. When you exercise, you produce more carbon dioxide gas, which dissolves with water to make a weak acid. You are now acidic. This causes a rise in heart rate so that you move more blood through the body, hopefully getting rid of the carbon dioxide in your lungs and picking up more oxygen. There are reflexes to compensate for your heart rate as well. The atrial reflex stimulus is a rise in venous return. That's the amount of blood entering your right atrium. Increased venous return causes a stretched right atrial myocardium. That stimulates atrial baroreceptors. This increases sympathetic nervous system stimulation and heart rate rises. A graphic of the atrial or Bainbridge reflex. Note how there is also an inhibitory reflex. This is an overview of the different mechanisms discussed. Work through this flowchart. Notice how all these methods work to change cardiac output by either changing heart rate or stroke volume. Methods that change heart rate: sympathetic nervous system, parasympathetic nervous system, epinephrine, excess calcium, increasing venous return. Methods that change stroke volume: contractility, changes in venous return, kidney activity by way of sodium and water reabsorption,